Okay. If you guys could do me a favor and sign in to this, just so people know that I'm doing my job. Because I know some of y'all can't make it to the study sessions or like the hours just don't work for you. So if you could sign in, I'd really appreciate it. Okay, I can get started. Um, I just wanted to go over the exam 3 review. And then just as a reminder, this is not a comprehensive exam. I mean, not a comprehensive review of the exam. Um, Highly recommend going through your notes and the lecture and also going through his kind of reviews and like study topics. Don't just solely use the review as like your only study material. Okay, that being said, you are studying how bacteria can change from a harmless form to a deadly form. You inject mice with different bacterial samples. So, harmless bacteria, the mice live. Deadly bacteria, the mice die, which is expected. Heat killed deadly bacteria, the mice live. And then harmless bacteria plus heat killed bacteria, the mice die. So after the last test, you find live deadly bacteria in the mice. So what does this suggest? So this, let's go through our answer choices. The heat killed bacteria came back to life and caused an infection. So that's wrong. If it's dead, it's dead. The harmless bacteria absorb genetic material from the heat killed bacteria. Okay, maybe. The mice were already sick before the experiment. That's wrong. Mice were fine. The heat killed bacteria released a toxin that killed the mice. No, because they, on one of the other things, they injected the deadly bacteria that was killed by heat and the mice still lived. So we know it's not that one. So your answer is B. And if you remember, this is the Griffith experiment where they did this experiment. But remember, you won't be tested on like what this experiment did and like who did what, but know how to like manipulate what was found from the experiment into like a real world application. So next you are studying how harmless bacteria can transform into a deadly form. You treat bacterial extracts with different enzymes that one breaks down DNA, one breaks down RNA, one breaks down proteins before mixing them with the harmless bacteria. So this is kind of piggybacking off the first experiment we were talking about, but now they're breaking down different components to see which component actually gives the bacteria the deadly form. So when DNA is not present, the bacteria no longer transform into the deadly form. What does this mean? So this means that when D, okay, when separating the deadly strains, DNA, RNA, and proteins, we find the causative agent of transformation is DNA. So and if you remember, this is the Avery, MacLeod, and McCarty experiment. This kind of just piggybacked off of Griffith's work. Okay, next you label the DNA of a bacteriophage with radioactive phosphorus and its protein coat with radioactive sulfur. After allowing the virus to infect the bacterial cells, you separate the bacterial cells from the empty viral coats using a centrifuge. If you only inject the bacterial cells without the viral coats, into a new culture of bacteria, which radioactive label would you expect to be present, and what does this indicate about genetic material? Into a new culture. So what you would expect to see is most of the radioactive DNA entered the cell and the proteins remained outside, proving that DNA is the genetic material and is what is uptaken from the environment. So, yeah, I'm just gonna say. Okay. And if you remember, that is the Hershey and Chase experiment. Okay. Now, DNA versus RNA, this is very important. Structure, DNA is usually double-stranded. RNA is single-stranded. The backbone, also very important. They both have phosphodiester bonds. And the same. If you remember, phosphodiester bonds are the bonds, so say this is DNA. That's it. It's A. And here's T. So here, where's my right here, the bond between the A and the T are hydrogen bonds. Remember that. And then here on the backbone is phosphodiester bonds. So remember that when we talk about uh base pairing versus like backbone pairing. And then sugar, also an important differentiation, is we have deoxyribose in DNA. And then we have just ribose in DNA. Hence the name RNA and then DNA, deoxyribose. Um, oh, and the differentiation is carbon 2 carbon 2 on the sugar ring does not have yeah. Go back on the slides and you'll see what I'm talking about. But remember that there's 5 carbons, and the only difference is that deoxyribose doesn't have a hydroxyl on carbon number 2. Okay, and then bases, which is also really important. DNA has A, T, C, and G. RNA has A, U, C, and G. And we have nuclear DNA and mitochondrial. And for RNA is located in the cytoplasm. Okay. Also really important, what's the difference between CG bonds and AT bonds? Which bases are purines and which are pyrimidines? So the difference between C and G bonds, A and T, is that A and T have 2 bonds. Well, yeah, 2 bonds, and C and G have 3 bonds. So C and G is well, he said it's 30% stronger. It's pretty stronger. And purines and pyrimidines. So our purines are A and G, and then our pyrimidines are C and G. Oh wait, should be C and T, right? Yeah. So our purines have 2 rings, yeah, and our pyrimidines have 1 ring. Okay, what's the difference between a nucleotide and a nucleoside? So a nucleoside is a base and a sugar. Is a base, a sugar, and a phosphate. And there can be multiple phosphates or just one, as long as it has one, but it can have multiple. That's what a nucleotide is. Okay. Which of the following is true about DNA nucleotide composition? So A's equals T's. No, because A binds to T. C equals G and C. Yeah, no, I said that wrong. A equals G and C equals T. That's wrong because, as we said before, A equals T and C is bonded to G. That one's wrong. A equals G plus T. That's completely wrong. And then we have the last one, G plus A equals T plus C, which is right because these like G, these are just switched basically. So this is just saying the amount of A is equal to the amount of T, and the amount of G's is equal to the amount of C's, which that is true because that's how our base pairing goes. So they have the base pairs on each side, which makes it equal. So that is our answer. Okay, what strand of DNA is a complement strand to the following? So be careful on your exam. Make sure you know the orientation of the thing, of your strand that you're picking, because the complement strand is going to be like the way DNA lines up, it's going to be like that. So T, A, A, okay, now let's see what answer choices line up. So it's going to be this one. It's going to be B because we start off with our two A's here. So we can take that one out and then we can take that one out. And then GC, are those the same? I think they are. Yeah. Okay, so it would be A and A or B. They're both right. I guess I duplicated it. Okay. So it's A and B. That's why I was confused. I was like, they're both white, right. Okay. It's A or B because they're the exact same strand. Okay. Moving on. Sorry about that. Which strand of RNA is the complement strand to blank? So it's asking you for RNA. So you're gonna do the same thing. Watch your primes. And remember, this is RNA. So A usually binds to T in DNA, but we're using RNA, so it's going to be U, A, C, G, A. Now we just have to look which one matches up, and this is B is going to be your answer. Okay. I was going to say I did the same thing on this one, but if you notice, 5 prime, 3 prime. So just be careful. Okay. DNA exists in a double helix structure. One characteristic of this structure includes blank. So parallel strands. Based on the questions we had before and what we know, DNA is anti-parallel because we have your 3s and your 5s. So anti-parallel. Next, counterclockwise direction orientation. You know, this is false. most DNA is clockwise. Okay, 10 bases per twist. We know that's right. Maybe let's go through all of our options. Purines pair with other purines. That's false. So purines are paired with pyrimidines. And 4.9 nanometers per complete twist. That's wrong because it's 10 bases per twist, and we know that. So our answer is 10 bases per twist. Okay, which alternate alternative double helixes is predominantly found in living cells and right-handed? So predominantly found in living cells is B-DNA. If you remember, Z-DNA is our zigzag one, and that one's not right-handed, that one's left-handed. Zigzag Z-DNA. And then cDNA doesn't even exist. I made it up. So what is RNA's tertiary structure and why is it important? So tertiary structure is the 3D shape. Okay, and why is this important? They carry amino acids at 3' end for translation, and so let me write that first. So the second reason is the anticodon region interacts with the mRNA and it must be exposed for it to happen. Chapter 12, Prokaryote versus Eukaryote location and structure. So prokaryotes have a nucleoid region and it is in direct our DNA, eukaryotes DNA is located inside the nucleus. Okay, moving on to structure. I'm just gonna write P and E. So prokaryotes are usually a singular circular chromosome. And we, as you know, we have sets of chromosomes. Um, and then in prokaryotes, protein encoding genes, structural genes account for a majority of their DNA versus, yeah. So a lot of their DNA is just structural genes. Oh, and we have multiple or origins of replication. Okay. What can induce supercoiling? Can you replace the DNA? Can you replicate the DNA with supercoiling? Um, so what can induce supercoiling is underlining and over. And can you replicate the DNA with supercoiling? No. DNA must be unwound. You have to decondense it because DNA is so condensed, you can't replicate it when it's all wrapped up. Okay. What does DNA topoisomerase do? One and two do. So one, two, one, if you remember, relaxes. And then topoisomerase two creates. Okay. So one relaxes negative supercoils and two creates negative supercoils, but also can relax. Well, topoisomerase 2 can relax positive supercoils as well. Um, what is transposition? Transposition, what are the differences, the different types of transposition? So transposition So when a segment of DNA is inserted into a new location, this is the part of DNA that's moved is called a transposon. And these are our jumping genes. And then we have simple and retro. And simple is our cut and paste. And then retro is our copy and paste. So cut and paste, you would just say you have a thing of DNA, right? Just a bad drawing. You cut this part out. Oh, I grabbed the whole thing. Okay, that's not gonna work the way I thought it did. Anyways, you're gonna cut this whole piece out and then move it, say, down here. That piece, you totally took it out and moved it. Versus retrotransposition, you're gonna have DNA, right? This is a piece that you're gonna take out. But retrotransposition, you're copying and pasting it. So it's copied and pasted over here. Or it could be pasted right here too. But the original is still there versus cut and paste, it's gone from the original spot. And this happens from the use of reverse transcriptase. Sounds familiar? Okay. Sorry for my bad drawing. But what is the order of eukaryotic chromatin compaction? So this is our a nucleosome to our 30 nanometer fiber to our loop. I don't know why I put that there. Loop domains. Okay. And then on this side, you would just have like I've seen sometimes they'll put like naked DNA here. And then on this side, they'll put like metaphase. So you might see it with all these, or you might just see it with like those. Either way, know both ways. Okay. Difference between euchromatin and heterochromatin. So euchromatin is transcriptionally over here. Heterochromatin is inactive, transcriptionally inactive. And euchromatin is less dense. Heterochromatin is more. Okay. And something that I was taught that still sticks with me is like euchromatin, you can use it. Heterochromatin, you can't use it. So like the U in euchromatin means you can use it. Something to remember. Okay, now we get into the nitty-gritty. So my suggestion would be for you to like have a blank copy like this and be able to name and give like the function of what each of these do. So first we're gonna name them. So, and then like you can also conceptually think of like what's happening as you're naming them. Here we have, I'm kind of gonna draw that to topoisomerase 2, which is also DNA gyrase. So, as we were talking about in class, its neighbor over here, helicase, is doing the unwinding, right? But topoisomerase 2 is helping relax the tension so that by the time it gets to helicase, like helicase has an easier time of separating and unwinding the strands. Okay, and then after helicase, our friends, I'm just SSBPs. These are our single-stranded binding proteins. These prevent our newly separated strands from coming back together because they're chemically like attracted to each other and they want to get right back to each other. So our single stranded binding proteins, like the name suggests, binds to a singular nucleotide and keeps it from binding to the other side. So we can keep it open. Here we have our DNA polymerase 3. This this guy is the star of the show. This is he's making it happen with the help of our other proteins, but he is synthesizing the new DNA. And then right here, important, these are our RNA primers. And what makes our RNA primers? Our primase. And then after that is again our star of the show, DNA polymerase 3. And then here we have an, I can never spell this, an Okazaki fragment. So if you notice, this is our leading and this is our lagging. Because this is getting synthesized all in one, no breaks, but here we have our primase synthesizing our primers and our Okazaki fragments. Okay. Oh, after that, because now that we're talking about our lagging strand, this is our ligase. Bringing back our, putting back DNA where the RNA replacing the RNA primers and putting back DNA. That's our friend DNA ligase. And yeah, so highly recommend going through all of these, knowing all of these, and know the function and what would happen if they were taken away. So let's say we didn't have single-stranded binding proteins. What would happen in the replication process? Well, the DNA would snap back together. We wouldn't have a replication fork, and DNA wouldn't have, like, we wouldn't get a new strand of DNA because it would be impossible because they would keep on sticking together. Or, like, what would happen to the lagging strand if we didn't have RNA primers? Like, we wouldn't be able to, we would have segments of DNA not. We wouldn't, I don't think we would be able to replicate even the lagging strand if we didn't have RNA primers. Because, yeah, I think we need the RNA primers to have the, yeah, because we need it to be synthesized from 5 to 3 prime, and we can't do that without the primers. Okay. What is induced fit and why is it important? So, induced fit is important because the DNA and this is important because of our proofreading. So if a pair is mismatched, it's removed using our exo. Our exonuclease. Okay, now, what would you expect a single, what would you expect an origin of replication to have a high percentage of? So we would expect an origin of replication to have a high percentage of A and T bond, oh, and T bonds because these there's only 2 hydrogen bonds versus G and C bonds have 3 and they're more stable. So an origin of replication, you want to make it easier for them to come apart. So you would have high percentage of AT bonds. What is the P pre-RC and how is it activated? So the P pre-RC is 6 proteins and it has a activated by phosphorylation. Okay. What polymerases are in eukaryotes and what do they synthesize? So in eukaryotes, we have alpha, beta, epsilon, which is that weird. Anyways, epsilon and gamma. These all synthesize nuclear DNA, and gamma synthesizes mitochondria. Okay, explain what flap endonucleases does conceptually. So flap endonucleases uh, removes primers. It removes small RNA flaps generated by DNA polymerase. Um, DNA polymerase elongates an Ozaki fragment until it runs into an RNA primer of an adjacent fragment. Okay, so okay. Why do we have telomerase and what is the function of telomerase? It's telomere. I said telomerase, right? Telomeres. So telomeres are the end pieces of DNA. And the reason we have them is every time we would do DNA replication, like the last bit would fall off. So we have telomeres. So if that happens, like it's nothing important. But if we didn't have them, like a bit of like the last bit of DNA would fall off every time like replication happened. So like we would end up with nothing after like a couple rounds of replication. So it's the end pieces of DNA, um, and then the enzyme, it maintains the length. So we can keep on like having that extra length at the end, so we're not taking any DNA that we shouldn't be. Okay, holiday model. The tunic separate the strands, and the break model is you have a double-stranded. Break. Okay. What are the stages of transcription? So we have initiation. Oh, okay. So obviously go into more detail than like what are the stages of transcription, but yeah, initiation, elongation, termination. Elongation, the RNA polymerase binds to DNA. The elongation, they synthesize the DNA, and then termination is obviously when the RNA synthesis stops. So how many bases are numbered relative to the promoter? So the answer to this is positive numbers for downstream and negative numbers for upstream. So say it starts here, upstream, and then you would have downstream. So these would be positive, and then these would be negative. How does RNA polymerase bind to DNA? So he said that this was pretty important and that we would come back to it, and now we're back to it. So, remember the major and minor grooves? He's like, when we're talking about it, he's like, it'll come back. Well, it's back. So RNA polymerase recognizes or binds to DNA by interacting with the major and minor grooves of DNA. So, in which direction is RNA synthesized? RNA synthesized, RNA is synthesized by the 5 to 3 prime direction. E. coli has two mechanisms for termination. What are they and their functions? So, our two mechanisms are row independent and row dependent. Um, row independent does not require the use of stem loop structures. Excuse me. And then our row dependent does require. Well, it requires a protein called row that functions as a helicase. Okay. In eukaryote organisms, how does alternative splicing impact protein production? So it enables the production of only one protein from a gene. That's not, it's like the opposite reason of why we have alternative splicing. So it results in the creation of a non-functional protein variant of a gene. So we don't want non-functional proteins, we want functional proteins. It allows the generation, it allows the, yeah, generation of multiple different proteins from a single gene. Yes, that is exactly the reason why we have alternative splicing, is we want different proteins from a single gene. Okay, which RNA modification plays a crucial role in maintaining mRNA stability in eukaryotic organisms. So, stability, yeah, let's think about like it's asking for stability. So, base modification doesn't have anything to do with stability. Alternative splicing also has nothing to do with stability. Processing also has nothing to do with stability, but poly-A tailing has everything to do with stability. It plays a crucial role in maintaining the stability of mRNA. And that is all I have for you. Good luck studying, and also remember that this is not comprehensive. This is just like key points that I think that are important. Um, if you have any questions, email me. I also highly recommend doing the past worksheets 3 and 4, I believe, to get some more studying in. But other than that, good luck on your exam.